Welcome to the Bodhi Academy. My name is Shrutika and today we will continue with our chapter Molecular Basis of Inheritance. So in this video what we will learn is which are the different experiments which were carried out to prove that DNA is the genetic material. So let us start with the video. So in search of the genetic material. So which was the first experiment which was carried out to understand that the DNA was the genetic material was the transforming principle. So this transforming principle so the transforming principle, it was in 1928. It was done by Frederick Griffith in a series of experiments. So what he did was, in his experiment, he took Streptococcus pneumonia, that is the bacterium possible for pneumonia, witnessed a miraculous transformation in the bacteria. So this was how he developed this transforming principle using the Streptococcus pneumonia bacteria which causes pneumonia in the organism. So when Streptococcus pneumonia bacteria are grown on a culture plate, some produces smooth shiny colonies that he termed as the S colony, while other produce rough colonies which he termed as the R colonies. So this is because S strain bacteria have a mucus, that is a polysaccharide coat, while R strain does not have the polysaccharide coat. What he did was he injected mice with S strain, that is the virulent strain, and what happened was the mice died due to pneumonia infection, but mice infected with R strain do not develop pneumonia. So he what he concluded was, if the mice was injected with R strain, that is the virulent strain, S strain, that is the virulent strain, the mice dies. If R strain is injected into mice, that is the non-virulent strain, the mice lives. Then Griffith was able to kill bacteria by heating them, and he observed that heat kill S strain bacteria injected in mice did not kill them. So S strain, that was S strain bacteria which were heat killed and then which were injected into the mice lived because there was no S strain. Then what he did was he took heat killed S strain bacteria with R strain live bacteria and injected into the mice. But here what happened was the mice died. Firstly, R strain bacteria, when it was injected, mice lived, and when heat killed S strain bacteria was injected, the mice also lived. But what happened over here, when S strain bacteria, which was heat killed, and there was R strain bacteria which did not cause the death of the mice, but here when this combination was injected into mice, what happened was mice died. So what he concluded was, when the so he concluded that the R strain bacteria has somehow been transformed by the heat kill S strain bacteria, and this was the transforming principle, wherein the transferred from the heat kill S strain had enabled the R strain to synthesize a smooth polysaccharide coat which became virulent. So what this heat killed S strain bacteria did was, it transformed the R strain bacteria into S strain bacteria. This must be due to the transfer of genetic material, and however the biochemical nature of genetic material was not identified from the experiment, but he concluded that this is the transformation which is taking place of the R strain into S strain, from non-virulent to virulent, and causing the death of the mice. So this is how the experiment was carried out, and that is why it was called as the transforming principle. Then what happened was the second experiment, which was a continuation of the transforming principle, which was done by Avery and McCarthy. In the genetic material they thought it to be a protein. They worked to determine the biochemical nature of the transforming principle in Griffith's experiment. They purified biochemicals, that is protein, DNA, RNA, etc., from the heat kill S strain to see which ones could transfer live R cells into S cells. They discovered that DNA alone from the S bacteria caused R bacteria to be transformed. So they also discovered that protein digesting enzyme, that is proteases, and RNA digesting enzyme, that is RNases, did not affect transformation. So the transforming substance was not a protein or RNA. Digestion with DNAs did not inhibit transformation, suggesting that DNA caused the transformation. So this is how it was identified that DNA is the hereditary material in organisms. Then the most important experiment, that is the genetic material, that proved that genetic material is DNA, and this was carried out by Hershey's and Chase in 1952. They worked with virus that infect bacteria called as bacteriophages. So Hershey and Chase worked to discover whether it was protein or DNA from the virus that enters the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some other on the medium that contained radioactive sulfur. So they grew viruses on a medium which consists of radioactive phosphorus and on some plates which consist of radioactive sulfur. Viruses grow in the presence of radioactive phosphorus containing radioactive DNA but no radioactive protein, because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain radioactive sulfur. Then radioactive phages were allowed to attach to the E. coli bacteria. Then as the infection proceeded, the virus coat were removed from the bacteria by agitating them in a blender through the process of centrifugation, and the bacteria which was infected with virus that has radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria, and bacteria that were infected with viruses that had radioactive protein were not radioactive. So this determined that the DNA carries the radioactivity. That indicates that the protein did not enter the bacteria from virus, but DNA is therefore the genetic material that passed from the virus to the bacteria. So this is how it was proved that DNA is the genetic material using the Hershey's and Chase experiment. So what are the properties of genetic material? So a molecule that can act as genetic material must fulfill the following criteria, that is, it should have the following criteria or the properties. It should be able to generate its replica, so that it should generate its copies. So replication is very important. It should be stable chemically and structurally. It should be chemically and structurally stable. It should provide the scope for slow changes, mutation, and are required for evolution. So it should cause slow changes that is required for the process of evolution. It should be able to express itself in form of Mendelian characters, which we have studied in our last chapter. So these are some properties which are important in the DNA and RNA. So now let us see the replication process. So what is replication? So replication is the term which was proposed by Watson and Crick after so this scheme suggests that the two strands would separate and act as a template for the synthesis of a new complementary strand after the completion of replication. Each DNA molecule should have one parent and one newly synthesized strand, and this is termed as semi-conservative DNA replication. So over here we can see that this is a parent strand of the double helix DNA. So what is replication? So replication is basically producing its new copies or producing new copies of the parent. So replication has to be like, it is semi-conservative. So why it is semi-conservative is, if this is the DNA, it will form a replication fork. From one strand of the DNA, there will be a new strand which will be synthesized, and this will form a second double helix DNA. Same way from here, another strand will act as a template, and then new nucleotides will be added and it will form a new double helix DNA. So this is how from one single parent DNA you will get two daughter DNAs. In this two daughter DNA, one strand of the DNA, double helix DNA will be the parent strand and one will be the newly synthesized strand. So that is why it is called a semi-conservative, because one strand of the parent is conserved in the new DNA strand. So this is the meaning of semi-conservative replication. So in today's class what we have learned is, what is semi-conservative DNA replication? We have seen the three models of experiments which determine that DNA is the genetic material, and we have also seen the criteria for a substance to be a genetic material. So thank you for watching today's video. Please don't forget to like, comment, share, subscribe to the Bodhi Academy.